Hai, Assalamualaikum dan salam sejahtera. In this video, I'm going to explain about human reproductive system, specifically the male reproductive system. Before that, let's review the male reproductive organ. So male reproductive organ basically involve penis, urethra, testicles, epididymis, vas deferens, prostate gland, semen vesicle, and bladder is the organs that contain the urine before it is being discharged through the urethra. We are going to look into detail where the spermatogenesis, the process of producing sperm, occurs. It is in the testicles. Now let's look at the spermatozoa structure. Spermatozoa is another name of sperm. The short name of spermatozoa is sperm. We usually call it sperm. So a spermatozoa must have a head, neck, midpiece, and tail. But basically, it involves three parts. The head, which is attached to the midpiece, and then attached to the tails. The neck is where the attachment between the head and the midpiece occur. So what should be in the head of the spermatozoa? The head should contain haploid nucleus, which has a tightly packed DNA material. Please remember, spermatozoa is a gamete, so the DNA, the amount of DNA in spermatozoa head should be half the amount of DNA of the parent cell. The head of the spermatozoa also contains a special vesicle at the tip of the sperm's head. It is called acrosome at the tip. Without this structure, it will prevent spermatozoa, the sperm, from fertilizing or penetrating the secondary oocyte. This is because acrosome contains hydrolytic enzymes to digest the membrane of the secondary oocyte. The digestion of the secondary oocyte membrane is very important to enable the sperm penetrating the egg, thus to fertilize. Without this action, without the penetration, without the digestion, fertilization will not occur. So the neck of the spermatozoa is a joining structure that joins the head and the midpiece. So this is because it contains centrioles. Centriole is to attach the sperm tail and the first division of the fertilized egg. Okay, this is the content of the DNA, the nucleus. The midpiece contain a large number of mitochondria. Sebab tu dalam struktur ni, bila dia lukis memang ada struktur mitochondria. This is important. Mitochondria are important to provide ATP for the tail, for the movement of the tail, because the tail needs the energy to propel, to enable the sperm to swim through the female reproductive tract to find the secondary oocyte in the fallopian tube. Okay, the tail is a flagellum. It's a flagellum with the 9+2 microtubules arrangement. So this tail aids in locomotion, propelling. The word propelling means it is spinning, producing a force that push the sperm forward. And so it propels the sperm forward. Macam konsep boat lah, boat yang ada engine tu akan berpusing. So the force produced by the propelling of the engine, of the fan, dia akan push the boat forward, sama macam konsep sperm. Let's look at the hormones that involve in spermatogenesis. Spermatogenesis is actually a process. It is a process that produces spermatozoa. Before males reach puberty dalam bahasa Melayu dia adalah akil baligh. Before male reach puberty, there is no spermatogenesis occur because lack of hormonal that trigger the body to start the spermatogenesis. So when male reach puberty, spermatogenesis will begin with the help of these 4 types of hormone. The first one is gonadotropin-releasing hormone, or also known as GnRH. So you can use the full name, or you are allowed to use the abbreviation, but it must be a correct writing. G mesti huruf besar, N kecil, R, H besar. Sama dengan the second hormone is follicle stimulating hormone, or known as FSH. Luteinizing hormone, also known as LH. And the last one is the testosterone. Later on, we will learn that there is another substance that is important during spermatogenesis. But for the time being, memorize these 4 hormones first. Testosterone is very related to male. Okay. Apa fungsi dia? Kita bincangkan nanti. So, gonadotropin releasing hormone, the GnRH, is secreted by the hypothalamus at the structure at the brain. Hypothalamus. Hypothalamus. This is the hypothalamus. This is the pituitary glands, which is important to secrete the other 2 hormones. GnRH is important to stimulate the anterior pituitary gland to secrete FSH and LH. So if the hypothalamus fail to secrete the gonadotropin releasing hormone, the anterior pituitary will not be stimulated to secrete FSH and LH. Okay? As we look at this diagram, the schematic diagram of hormone relationship, we can see that hypothalamus secrete GnRH. GnRH will stimulate. The word is stimulate. Stimulate anterior pituitary to secrete FSH and LH. Okay, what does FSH do? From this diagram, it shows that FSH will act on Sertoli cells, while LH will act on a different cell, which is the Leydig cell. Okay, then what happens to the Sertoli cell? Sertoli cells do something about inhibin, and then it involves in spermatogenesis. So Sertoli cells secrete inhibin, while another involvement is Sertoli cells triggered to undergo spermatogenesis ataupun assist in, okay, the correct word is assist in spermatogenesis. So this is when the sperm is produced. So without the assistance of Sertoli cells, the production of spermatozoa or sperms might not occur, or it would be not in the correct order. FSH secreted by anterior pituitary gland to stimulate Sertoli cells. So what does Sertoli cells do? Provide nutrients for the development of spermatid into spermatozoa in tubule. So this is a parts of spermatogenesis. Another function of Sertoli cells is to secrete inhibin that is important to inhibit FSH. Sebab tu ada anak panah merah ni, negative feedback. Meaning, a high concentration of inhibin or the secretion of inhibin will eventually inhibit anterior pituitary from releasing another FSH. So production of FSH will be stopped. So Sertoli cell will not be stimulated to help in spermatogenesis. Jadi dia sepatutnya cycle ataupun kitaran untuk menghasilkan sperm dia akan berhenti sebentar. The LH, luteinizing hormone, is secreted by anterior pituitary gland. Sama But the role of hormone is to stimulate a different cell, the Leydig cells ataupun interstitial cells. So what is the difference between Leydig and Sertoli cell? Kalau kita tengok gambar rajah ni, okay, this is seminiferous tubule. This is testicles. If we cut across the testicle, and then we will come across this structure. Consists of a few seminiferous tubules. This is seminiferous tubule. Inside the seminiferous tubule, this is the seminiferous tubule. Inside the seminiferous tubule is the primordial germ cells and Sertoli cell. So Sertoli cell is in the seminiferous tubule, while Leydig cell is at the interstitial in the area in between the seminiferous tubule. Maknanya dia dekat luar daripada seminiferous tubule. So Leydig cell is important to secrete testosterone. This is another name of Leydig cell, interstitial. Sebab dia duduk dekat interstitial area and in between the seminiferous tubule. Leydig cells is to secrete testosterone and other androgen to promote spermatogenesis. So spermatogenesis ni dia ada dua hala punya stimulation. One is from Sertoli cells, another one is from Leydig cells. Leydig cell daripada testosterone lah. Jadi testosterone is to promote spermatogenesis and also development of primary and secondary sex characteristics. That's why when male reach puberty, there are a few more characteristics that will be enhanced, like having stubble, mustache, beard. They become more hairy and more muscular. The formation of muscles increase. Jadi kalau dia bersukan, dia akan nampak lebih muscular. This is a testis structure, the testicle. This is what I've told you before. In the testicles, there are so many seminiferous tubule, and in the tubule there will be primordial germ cell that will then develop into a primary spermatocyte and then into a sperm. Okay, into a sperm. Okay. In between the primordial germ cells, there are Sertoli cells. Sertoli cells is important to provide nutrients for the growing spermatozoa. While the Leydig cell is at the outside of the seminiferous tubule in the testicles. It secretes the testosterone. Testosterone are hormone that stimulates spermatogenesis by stimulating growth and development of germinal epithelial. Jadi ini sel awal yang nak membentuk the germ cell yang nak membentuk gamete. It also stimulates Sertoli cells when work with FSH. Along with FSH, testosterone ni dia boleh stimulate Sertoli cells. And also it will regulate the level of other hormone through negative feedback mechanism. Sebab apa? Nampak anak panah ni. Okay, anak panah ni menunjukkan higher levels of testosterone will inhibit first the anterior pituitary. So LH will not be secreted. Another effect is towards hypothalamus. So the high level of testosterone will inhibit production of GnRH. So at this time, the effect of testosterone and both testosterone and inhibin will stop the spermatogenesis for a while. This is to give time for the for the sperms that is being produced in the cycle before to be released by the male. Inhibin secreted by Sertoli cells is to inhibit anterior pituitary from secreting FSH. Okay, if rates of spermatogenesis is too low, inhibin will not be secreted. Maksudnya spermatogenesis ni rate dia maknanya dah banyak dah sperms terhasil. Jadi they need to slow down. Kalau tidak nanti nak kena akan banyak sperm terhasil dalam seminiferous tubule. It needs to be ejaculated or released. Keluar ke epididymis and then released through the urethra. Negative feedback mechanism in spermatogenesis. This is the schematic diagram that shows the relationship between the tadi 4 kan? 1, 2, 3, 4. Sekarang kita tambah satu lagi iaitu inhibin. So 5. Jadi ada 2 yang melibatkan negative feedback. Okay. So if you were asked to explain negative feedback mechanism in spermatogenesis, high level of testosterone. Sebab ni negative feedback, kita akan tengok pada yang ini, negative feedback. First, testosterone, high level, mesti sebut high level. High level of testosterone will inhibit the hypothalamus and anterior pituitary. Thus, GnRH. Jadi, mula-mula dia akan inhibit hypothalamus. So, no GnRH. No GnRH. So, no GnRH anterior pituitary tak boleh rembeskan LH dan juga FSH. Sebab tadi inhibin dia negative feedback towards anterior pituitary. Kemungkinan hypothalamus masih lagi active in secreting the GnRH. So, testosterone is making it definite that hypothalamus will stop releasing GnRH. So anterior pituitary will not secrete FSH and LH. If spermatogenesis proceeds too rapidly, Sertoli cell will release inhibin that act as inhibitor towards anterior pituitary glands from secreting FSH. Inhibin may also have a slight effect on the hypothalamus, thus will reduce GnRH secretion. So when the rate of the spermatogenesis is low, inhibin will not be secreted. Thus it will stimulate FSH and LH production. Spermatogenesis is the process of formation and development of sperm. Occur in seminiferous tubule of testis. Let's look at the terms used here. Primordial germ cell or germinal epithelial cell. So it is actually an epithelial cell in the seminiferous tubule. So when it develops into a spermatocyte later on, it will continue undergoing meiosis and mitosis and differentiated into mature sperms. So this germinal epithelial cell, or you can use primordial germ cell, divides by mitosis. Dia akan membahagi dulu supaya bilangan dia banyak. Sebab apa? Kalau dia tak membahagi, kalau satu tu terus undergo meiosis, dia dah akan tak ada lagi sel yang lain yang nak meneruskan the next cycle of spermatogenesis. So they need to increase their number, and some of the daughter cell will then develop into a cells that can continue with the spermatogenesis. So the spermatogonial stem cell will divide repeatedly, another division of mitosis, to produce spermatogonia. So one type of spermatogonia will remain at the basal membrane. This is basal membrane. Jadi dia akan ada yang tinggal. Ni tinggal Yang ni akan differentiate and enlarge more. And then it will undergo meiosis. Kalau semua undergo meiosis and produce sperm, siapa yang tinggal untuk the next cycle? Itu maksud dia. Another spermatogonium will divide by mitosis to enlarge and generate primary spermatocyte. So nak mula spermatogenesis, dia akan mula dengan primary spermatocyte. Yang lain semua adalah sel-sel yang asas. Nak membentuk primary spermatocyte ni, dia perlu bahagi banyak kali supaya ada sel yang tinggal untuk meneruskan spermatogenesis yang seterusnya. So spermatogonium, dengan ringkasnya, spermatogonium akan bermitosis dan kemudian differentiated, membeza kepada primary spermatocyte. Spermatocyte primer ataupun primary spermatocyte. So, starting from this one, this is the steps of spermatogenesis. Walaupun yang ni pun sebenarnya spermatogenesis juga, dan penghasilkan spermatogonia, tapi dia lebih kepada cell division that increase their number. Spermatocyte, which is diploid, will undergo meiosis 1 to form 2 haploid secondary spermatocyte. Primary akan membentuk secondary. Primary membentuk secondary. Primary undergo meiosis 1 and produce secondary spermatocyte. So, the secondary spermatocyte is already haploid. Sebab kita tahu dalam cell division, meiosis 1 produce, after meiosis 1, the daughter cells should be haploid. Each secondary spermatocyte continue with meiosis 2 to form 2 haploid spermatid. Jadi, in the end akan ada setiap satu primary spermatocyte akan hasilkan 4 spermatids. Satu sel, satu primary spermatocyte, 4 spermatid. So, spermatid is still non-motile. Maknanya tak boleh bergerak. Dia tak boleh release lagi. Kalau release spermatid, maknanya sperm itu masih belum tak sesuai. Dia tak boleh berenang sebab dia non-motile lagi. And still embedded in the Sertoli cells to gain nourishment. Each spermatid undergoes spermiogenesis to form spermatozoon. So what is spermiogenesis? Spermiogenesis is the process of preparing the sperms to form spermatozoon. Jadi bila dah jadi spermatozoon, it should be a sperm that can swim. So one primary spermatocyte can form 4 spermatozoa. So this is spermiogenesis, is a differentiation. Dia pembezaan, dia nak bezakan dengan kemudian dia enhance the structure of the sperms to make it better, to make it more viable. So after spermiogenesis, spermatids become more elongated, and then they also shed some of the cytoplasm, excessive cytoplasm, and develop a chromosome and tail.